I would like to open this discussion with three thoughts I would like to introduce here. One is about the context sensitivity of risk assessment. And the second point I would like to make is about the new dual-use dilemma. And the third point is what I always talk about. It's about risk communication and crisis communication. And to start with my first point about the context sensitivity of risk assessment, I brought you something with me. So I would like to take you to the 1990s. And thanks to climate change, researchers could make a new discovery. They discovered a virus in the body, in the eyes, and since it gets warmer, so they could isolate a virus in this body. And this virus is an influenza virus, the influenza virus of the 1918 influenza. And they isolated this virus, and they reconstructed it and said, voila, look, H1N1, wonderful, we have this here. This was in the 90s. Then we had 9/11, and everything got very, very, very threaty. So research on influenza virus was dangerous, yeah? So no way to do this. It was dangerous. Because they thought, if you can reconstruct and build this virus, you can spread it out. You can use it as a bioweapon. So no good at this. And then 2009, exactly the same virus came by, H1N1, and high popularity again. Good that we had this research. So the point with this, it's a bird, it's not a swine. So I'm triggering my next point with my avian influenza, but I'm glad you got it. The point is, the risk assessment depends on the context in which you make this assessment. 10 years ago, it was a high threat to work on this. 20 years ago, it was a good opportunity. Great. And nowadays, it's a bit ambivalent. Why is this ambivalent? So I would like to come to my next point, and I put the bird back again. So my next point is about the dual-use dilemma. And I would like to take you, I mean, you're all, I think, familiar with this. Dual use is a difficult decision whether something is good or bad. Yeah? And I would like to demonstrate this with my toys here. This is a firefighter. Yeah? And you know exactly what he is doing and what he's about. So it's very, nothing ambiguous about this. So a firefighter can do things. And this is basically the one use. Dual use is when things getting a bit ambivalent. So Lego building blocks. Yeah, you can build different things with it, with the same blocks. You can build a racing car, or an ambulance, or a tank. Yeah, the same blocks. And this is the particularity of biomedical sciences. They do only these building blocks. They don't do these firefighters any longer, where you can tell this is bad or good, depending on what they do. And this is a new challenge, because it's not about the material. You can get these building blocks everywhere, yeah. And you have different colors. You can ask scientists, it's a red block, it's a yellow block, that's fine. It's about the way you put them together and the knowledge to do this. So dual use has developed a different layer, a different dimension. It's a new dual use because it's information sensitive. It's the information and not the material itself. So here we are with this context sensitivity of risk assessment. And then we have this new dual use dilemma where sort of everything is about the information you share in an information society. So what do you do about this? So you do a lot of communication. But to be honest, you do a lot of this here, firefighting, crisis communication. You start communicating when it's too late, yeah? When you come to distinct a fire. And I would suggest to go to a risk communication attitude, to talk about the different ways you can assemble a car, an ambulance, a tank, to talk more broadly, involve more people about these things. And this is the risk communication I would like to encourage in this debate about risk assessment and dual use. Because at the moment, I feel that we have pretty much ad hoc communication, like this firefighting crisis communication, what to do now. And we feel that we had this mutant bird flu discussion when engineers, biologists engineered a new influenza virus, a mutant one. And they said, well, it's dangerous, you shouldn't do this. And the reflex was to stop the virus, the scientists do this. Yeah? Our reflex is to limit and restrict this because we feel it's dangerous from our current risk assessment. And I would like to encourage broader risk communication about these aspects and not this ad hoc firefighting. And this involves a broader perspective. It's not just the scientific perspective, and I think this is the reason why we're all here. It is immensely a political, economic dimension in these areas, and we can't tell exactly what is good and what is bad, and it's up to a society to decide on this. And this is what I would like to encourage, a risk communication discussion in a broader dimension.
I'm a political sociologist, and like Petra, I'm also interested in risk. And particularly, I'm not so interested in the risk communication aspect. What my interest is more focused on is the way in which certain risks become things that we focus on or become problems that we then decide to respond to. So in the words of the French philosopher Michel Foucault, what I'm interested in is this problem of, or the process of problematization, or in the words of the American anthropologist Mary Douglas, I'm interested in how certain risks come to be placed higher in the risk portfolios than, for example, other risks, because I start from the starting point that there's nothing inevitable that says certain risks, that there is a risk hierarchy. I start from the premise that it's contextually based. So for me, the interesting question to ask today is about how the threat or the risk of bioterrorism has emerged over time. And that's what I will talk a little bit about in the next few minutes. Bioterrorism is a relatively new concept. Of course, the idea of, or the biological weapons have a longer history themselves. It stretches back to the interim between the two world wars, when they started being developed in state programs and tested. And since that time, there have been international treaties that Nigel referred to in his opening remarks, the Geneva Protocol, the Biological Weapons Convention, that ban their use, that ban their development, production, and stockpiling. And the use of biological weapons was for a long time sort of held in check by nuclear weapons, by the threat from nuclear weapons, during the Cold War, by these international treaties. But at the end of the Cold War, something happened. A new security threat came to be identified by security analysts in the United States, particularly those on the right of the political spectrum with ties to the Pentagon. And this new threat was terrorists linked with weapons of mass destruction, and that included bioterrorism. If you analyze early political discussions on bioterrorism, you will see that there were many different perspectives about the risks, about the currency of the threat, about the possibility of their use, about the imminence of their use. And you can broadly divide the people speaking about this into two groups. On the one hand, you'd have alarmists who would emphasize in their discussions the vulnerability of populations, who talk about apocalyptic scenarios or apocalyptics, attacks where you would see the deaths of thousands of people with exceptional consequences. On the other hand, you would have skeptics who would sort of question this scenario and who focused their discussions around bioterrorism less on those sort of technical things about what was possible and talked more about what was probable, or they focused more on the intention on who were the terrorists, what were their intentions, what were their capabilities, what were their interests in using biological weapons. So you'd have these two groups, and as this debate was playing out, you could analyze the different places, the different events, the different characters that in the end resulted in the alarmists triumphing over the skeptics and money pouring into biodefense programs. And I'm sure you will all remember a number of the events that happened around the time. This was in the 1990s. These debates were primarily taking place in the United States. Some of the events were the Shinrikyo attacks on the Tokyo underground that were with sarin, not with biological weapons, but the Shinrikyo group was also developing or trying to develop anthrax with little success, but that was still it was still a key event that was very much influencing the debates at the time. And other events, the Iraq and the weapons of mass destruction that were found in Iraq, the discovery of the enormous Soviet biodefense or offensive bioweapons program that they had there. Some of the key actors or the central actors that were taking part in the debate and that pushed the kind of the alarmist strand were the executive and the legislative branches, prominent members of the scientific security communities, biotechnology communities, as well as the media. And the media was certainly a place where some of these debates played out. I'm sure you'll all remember Defense Secretary Dan Cohen very publicly saying on ABC News, holding up a bag of, a 5-pound bag of sugar, saying, if this was, I can see some of you nodding, if this was anthrax and it was released over Washington, half of the population would die. There were also a number of scary novels around this time. The Cobra event, for example, is often one that people remember. But these debates were also taking place, of course, in government settings, between departments and testimonies and hearings, in academic articles and tabletop exercises, etc. Then 9/11 came along and the anthrax letters that followed, and the attention paid to bioterrorism increased dramatically. The idea of bioterrorism as super terrorism was spread then from Washington to security communities around the world and back to capitals from those different security communities. This idea of super terrorism had the effect of thinking about bioterrorism as super terrorism, sort of has the effect of empowering certain groups of people. So people in war, people in defense, people in international order and strategy were empowered by thinking about bioterrorism like that. Also other groups connected with crime, connected with internal security, with public order and police investigations were empowered. And in the last minute or so that I have, I just want to focus on how we think about bioterrorism today, because there has been a real shift in how we think about bioterrorism today. And along with that shift comes a different way of responding to bioterrorism. a different way of intervening with bioterrorism. Today, bioterrorism is no longer thought about as this one standalone threat. Bioterrorism is thought about as one element of a spectrum of disease threats that also include natural outbreaks of disease. There's a very clear link these days between security and health. Other elements of this spectrum are unintended releases, which Laurie might be talking about later on, I imagine. Also negligence, also sabotage, unintended consequences of laboratory works, which I suppose you touched on a little bit, Petra, in your talk as well. And this way of thinking about bioterrorism has, as I said, consequences for how we think about how we should respond to bioterrorism. And groups that are now being empowered are those related to healthcare, to medicine, and so on. So I think I'll leave it there for now.
Since Petra showed us a prop, I'll be mostly talking about a lot of things that some of you have probably read already, so forgive me if it's repetitive to something that you've already read. Main messages, we've gone from an era where we were looking at controlling substances, controlling key microbes, special pathogen lists. Somehow everybody was supposed to keep track of who had test tubes of what. That is all completely irrelevant now. We're now in an information age, and we're not doing a good job of tracking information in any context, much less in the context of biology. We're in 3 great revolutions in biology right now that are unfolding at lightning pace and are completely turning biological research upside down. And they're all made possible by the plummeting cost of genomic sequencing. So, first human genome was sequenced. It took 10 years, 160 laboratories, a net cost of well over $10 billion if you combine the public and private sector costs. And today, most scientists in the rich world consider the actual sequencing costs to be pretty trivial in their budget requests for biology research. We're now down to full human genome sequence can be done for about $900. And the latest biotechnology magazine forecast is by 2016, that's going to have fallen all the way into the under $100 range. And you can buy a home sequencer, do your own sequencing, sequence your own genome overnight. The sequencer itself right now will cost you about $5,000, but there's newly advertised products coming out later this year. They're going to take it down into the 2,000 range. I'm ready to predict that it's going to defy Moore's law on the computer side. All of this is just plummeting at a dramatic pace. And this means, in fact, Craig Ventnor just announced last week that he's creating a whole new subsidiary company with the goal of sequencing 10,000 human genomes a year and doing complete analysis of relevant sequence genes, and with the goal of ramping up to 40,000 a year and creating the ultimate human genome database. Well, it's all gotten so cheap that we're outsourcing most genomic sequencing to one single place, the Beijing Genomics Institute, which you can decide if you think this is a good idea or not, but it now has the world's largest genomic repository of information, larger than any government repository, except to the degree that nothing in China is completely outside of Chinese government influence or control. So this incredible decrease in the cost of actual sequencing has made 3 things possible. I'll touch on them briefly. First, metagenomics. Second, synthetic biology, and finally, gain-of-function research. So metagenomics, we're now shotgun sequencing the entire world. We're shotgun sequencing everything around you, everything you breathe, everything you eat, everything inside of you. In the 1990s, for example, marine biologists were capable of telling you about a handful of microbes and what role they played in the ocean system, the general ocean ecology. That started to transform in the early 2000s with some key genomic work, but it was still slow, it was expensive, we didn't know much. In the last 5 years, the field has exploded to the degree that we now recognize that there are 100 million microbes per square centimeter in the oceans, which, by the way, it's about 1 million per square centimeter inside your body. And the computer systems, mass computationals, all over the world cannot keep up, cannot possibly begin to digest the information that's pouring in. We're looking at about 10 to the 29th microbes in the oceans, and the numbers of species involved is so vast that we're discovering entire phyla are playing a role that 10 years ago nobody even really knew about. And by the way, that's true inside your body. We're now discovering that your body will not work without archaea. Who knew archaea were inside the human body? They were called archaea because arcane, old, ancient. We didn't think they played any particular vital role in modern contemporary life forms. And indeed, you cannot live without them inside your body. So we're sequencing, we're learning, it's exploding. And in the process, of course, what's the first target a biologist wants to know about when they look at a new microbial species that they have sequenced? What are the virulence genes? What are the genes associated with transmissibility? What makes this microbe capable of infecting this range of species out there? What would it take to tweak it and make it infect something else? Or transmit this virulence or toxin to something else? This database is building at lightning speed, which leads to the second great revolution. You use Legos. I love that because a lot of people in synthetic biology talk about biobricks, which are often seen as analogous to building with Legos. You can now order which oligonucleotide sequence you're interested in. What DNA do you want to swap, build, pile around? Jerry-rig as you will. Create microbes in your lab, previously non-existent life forms. We now have teenage competition that involves about 38 countries in the world, where the goal of the competition is to create a previously non-existent life form. And the best new life form wins the prize. If you don't know about all this, you need to talk to your teenagers because they do. And this, of course, has resulted in Gerald Joyce famously referring to the new era of directed evolution. No longer are biologists sitting back observing life and trying to assess whether if you throw in an antibiotic, the life form changes in a test tube. Now one is directing the evolution of the life form, altering it to determine what its functionality may be and what its risks may be. And this allows you to construct whatever you may, and it means the information itself. What are the genetic sequences? What are the virulence factors, and so on? That's the key now that requires observation and regulation, not the microbe itself. Now you get to the whole question of gain-of-function research, which sometimes overlaps with what I've been describing and sometimes is actually using kind of old-fashioned forms of biological research. Gain-of-function, so the first idea of gain-of-function comes from Fritz Haber, who famously in Germany before World War I developed a way to do nitrogen fixation as a fertilizing system, but the same capacity allowed you to create chemical warfare. And Fritz actually so thoroughly embraced the duality that he performed personally both forms of research and was a supplier of chemical weapons for the German army in World War I. That's the most dramatic example of gain-of-function. But in most cases, when we talk about gain-of-function dual-use research, we're really talking about fairly innocent intent or even highfalutin intent, save the world intent. The most dramatic, of course, came forward when Ron Fouchier stood up at Malta in September 2011 and announced to a scientific meeting of European virologists, quote, "I did a very stupid thing," end quote, and then proceeded to describe how he had altered the H5N1 bird flu to turn it into a mammalian transmissible form of the bird flu virus. Given that H5N1 has a mortality rate in Homo sapiens infected to date of 61%, that's a very frightening possibility that one would deliberately alter this normally only bird-to-human virus to be a potential human-to-human virus. And so with that having been performed or announced, Yoshi Kawaoka at the University of Wisconsin, Fouchier, by the way, is at the at Erasmus in Rotterdam. Yoshi Kawaoka said, well, I've done a very similar experiment, and I think it's a smart thing to do because we're trying to come up with vaccines in anticipation of a natural occurrence, an evolution. Again, we're talking directed evolution here. Of course, one big difference between Fouchier and Kawaoka was Kawaoka deliberately crippled the virus he was experimenting on so that it couldn't actually escape the lab, or if it did, it couldn't actually harm human beings. Fouchier did not. This has opened up this huge kettle of fish, very stinky fish, really, and gigantic controversy on both sides of the Atlantic, and in fact, also in the Pacific. On our side, the US, it brought all sorts of previously unknown committees forth, all sorts of responses from obscure entities saying you can or cannot publish this, you can or cannot explain this to other people, this is or is not evil, terrible, wonderful, public health, whatever. In Europe, this response was slower, but when it came, it was perhaps more surprising. Ron Fouchier had already published his work in Nature. Yoshi Kawaoka published his after long debate in the United States in Science, including materials and methods, which was the question. Should you, simply to avoid a dual-use catastrophe, tell everybody not to publish materials and methods? Which I said was absurd because already 18 postdocs have emailed it to 18 friends, which have then been emailed to 45,000 friends, and it's utterly irrelevant what is physically published. At any rate, Fouchier's work belatedly, or however you want to look at it, was ruled by a Dutch court to have violated European export control law by publishing the how-to. And it is still under close scrutiny, quite a divided issue here in Europe, with some European scientific groups saying that's absurd, it'll slow down research, it'll put Europe at a disadvantage in the general race to invent and innovate, and others saying absolutely, none of this should ever get published and we should condemn Erasmus University for their behavior and so on. This is yet to be ultimately sorted out. And just to quickly get to a wrap up here, as I see the clock ticking, we're now at a situation where, all right, the issue is information. But all legal apparatuses that exist, whether you're talking about the Biological Weapons Convention, the Cartagena Protocol, individual national legislation, or the international health regulations, pertain to specific microbes. So they are, shall we say, physical, as opposed to information being ephemeral. They are, you can say this glass has, pick one. Let's say H5N1 in it. But you cannot control or know who is sending information about sequences of H5N1 genes. Now that the Harbin laboratory in China sequenced or made 127 previously non-existent forms of H5N1, 5 of which transmitted readily through the air between guinea pigs, causing disease, the genie is out of the bottle here. But we don't have any system in place for really talking about how to regulate the flow of information, or even concretely of biobrick sales, and of who has access to what oligonucleotides to construct what. My greatest concern actually is less about bioterrorism per se, some nefarious individual or group constructing some evil, horrible microbe, but stupidity, mistakes, the zealous pace of the research, the fact that everybody is sloppily trying to do everything all at once, that everybody sees metagenomic shotgun methods as the way to go. And I would just simply remind you that the last case of smallpox was not in Nature, it was at Porton Down. And the last outbreak of SARS did not naturally arise. It occurred in 2005 out of the CDC laboratory run by the Chinese CDC in Beijing, and got out of the lab into the community. So last time I looked, we'd had a total proliferation of biosafety level 3 and safety level 4 laboratories, and a proliferation of accidents associated with those laboratories. This is what worries me. Finally, we have no regulation of the private sector whatsoever. So who knows what's going on at your local biotech laboratory or pharmaceutical plant.